Hello all, thank you very much for joining us today to Gilbert Kahana webinars. Today we're hosting Gmida-Sales Management with Julian Adams as the CEO, with Michelle Corfin as the Chief Operating and Commercial Officer, and Shai Lankri, the CFO of the company. We'll start as always with the company's presentation. Julian, please go ahead.
Thank you, Moshe. It's a pleasure to be here today and to address our investor audience. At Gmida, you can see that we are inspired to cure. Let me begin with our disclaimers, and you can consult our SEC filings for further details on forward-looking statements. At Gmida, we are committed to cures. I've been working for almost 40 years in R&D and developing medicines for patients, and in the last 5 years, we're seeing a revolution in medicine, specifically in immuno-oncology, where cell therapies are beginning to cure patients. And Gmida is at the forefront of this revolutionary period in medicine, and we would like to highlight our company's progress and be part of this community. We have a very experienced management team and leadership team with over 200 years of experience in discovery, development, and commercialization. And I'm joined here today with two of my colleagues, Michelle Corfin, our Chief Operating and Chief Commercial Officer, and Shai Lankri, our Chief Financial Officer. At Gmida, we are advancing two very promising clinical candidates that are leveraging our unique cell expansion technology platform. We're making great progress on Meduvacell and are preparing for BLA submission at the end of this year. We have the potential to be the first FDA-approved product for hematopoietic stem cell transplant, for bone marrow transplant. Our second program, which is in the clinic, is called GDA201. This is our natural killer cell or NK cell product, which is going to enter an important phase 1/2 trial in Non-Hodgkin Lymphoma in the second half of this year. And we have had a very successful phase 1 program that I'm going to share data with you today. And finally, we have a very strong financial position. We finished the first quarter with $175 million in the bank, which gives us cash into the second half of 2022. The basis of our technology is shown here. This is called the NAM platform technology, which allows us to expand cells, blood cells of any type, to increase the number of donor cells for patients. And the key to this technology is not only do we see robust expansion, but we preserve the function and potency of those cells after the expansion process. And as I've mentioned, we can expand any cell type in the body, stem cells for bone marrow transplantation, or as you will see, natural killer cells for lymphoma. Shown here is our pipeline, where we've NAM-enabled all of these opportunities. Our most advanced program is Meduvacell, which has completed a phase 3 trial, and I'll share the data with you in a moment. But I'd like to point out that we have both FDA breakthrough designation as well as orphan drug status. And this puts us in a very unique position with the FDA to get frequent contact, frequent scientific advice, and support from FDA on the way to registration. We have an additional program, a pilot program in a non-malignant blood disorder called severe aplastic anemia, and we have successfully transplanted 9 patients, and we have some of those patients disease-free out to 3 years, which is a cure. Our second program is our NK program, our natural killer cell program. The first product candidate is GDA201. And we began our first trial with fresh cells produced at the University of Minnesota. And I'll share clinical data from that trial that gave us the proof of concept. Today we have optimized the manufacturing process. We have a cryopreserved product which will enter the clinic very shortly for a phase 1/2 clinical trial in combination with rituximab, and this will be an off-the-shelf product to support a multi-center trial. Behind GDA201, we have GDA301, GDA401, and GDA501. These are genetically engineered cells to make them more potent and more attractive with a greater potential to also approach the treatment of solid tumors. So let's begin with Meduvacell, a potentially curative treatment for patients who need a bone marrow transplant. Meet Stacy. She inspires us all. She was diagnosed with acute leukemia 10 years ago and couldn't find an adult match in the registry. So she was referred to Duke Medical Center for a clinical trial, and she was one of our first patients treated with Meduvacell. She's 10 years from her transplant, disease-free. This is what a cure looks like. And by the way, this is what I saw when I joined Gamida-Sel and inspired me and the rest of the company to produce more Stacys. The key, again, to our technology is the ability to expand cells. Our starting material is umbilical cord blood. These are the youngest cells, the most pristine and potent cells to begin a bone marrow transplant. Umbilical cord blood cells have been used for almost 30 years. Because of the naive nature of these cells, we don't need a perfect genetic match. So by starting with umbilical cord blood, we have 99% certainty to find a match for everybody. The only limitation of cord blood is the dose. Based on a newborn's number of cells, it's insufficient dose for an optimal transplant in adults. So Meduvacell solves that problem. So the umbilical cord blood comes from cord blood banks all over the world and can be shipped to our manufacturing site. You can see that we separate the stem cells and expand them in culture for 3 weeks with NAM and growth factors. We also preserve the non-cultured fraction, which contains these naive T cells, which helps not only with engraftment, but long-term immune reconstitution. So a bone marrow transplant is really resetting the entire immune system with a healthy immune system with Meduvacell. Prior to our phase 3 studies, we spent years perfecting and exploring Meduvacell in smaller trials. We did phase 1 and phase 2 trials, and this gave us confidence to proceed with a pivotal randomized study. And at the same time, we got FDA breakthrough designation, and so we collaboratively with the FDA and with the EMA, the European authorities, to construct this global pivotal study which would support registration. You can see here we are enrolling patients ages 12 to 65, including adolescents, patients with serious hematologic malignancies, including high-grade leukemias and lymphomas. All patients were eligible for transplant in that they were in remission, but at high risk for relapse. So they needed a bone marrow transplant to survive. And all of our patients in our trial could not find a matched donor in the registry. We randomized patients to Meduvacell versus standard cord blood as the comparator. The endpoints included the primary endpoint, the time to neutrophil or white cell engraftment, which is the key infection-fighting cell. And then the secondary endpoints included platelet engraftment, which is another type of blood cell, reduction in hospitalization, and reduction in infections. And I will show you the data momentarily. We have additional exploratory endpoints related to safety and survival. So we randomized 125 patients at 33 sites around the world on 4 continents. The demographics and patient characteristics were well balanced in both arms, and Meduvacell was generally well tolerated. As you can see here, we met our primary endpoint. The time to neutrophil engraftment of median time was 12 days versus 22 days, with a very statistical significant p-value of less than 0.001. So these were all patients randomized, which is called intent to treat analysis, which removes all bias in the analysis. However, if we looked at the patients who actually were not only randomized but made it to transplant, we now have 108 patients. So there was a reduction in both arms of the study, patients who relapsed or patients who became ill or developed a fever and became ineligible for transplant. So this is the per protocol treatment. And now we see the same statistical significance, but now the time to engraftment was 10 days, which is a world record. If you compare Meduvacell to all other modalities of transplant, including matched-related donors, a sibling donor, a matched unrelated donor, a mismatched donor, or a haplo donor, which is a half-matched donor, we are at least a week or more superior to any other modality of transplant. We met also all the secondary endpoints with statistical significance. The time to platelet engraftment, an important white blood cell that controls the clotting factors. We reduced time in hospital by about 12 days. And very importantly, we reduced significantly bacterial, fungal, and viral infections. So let me turn it to Michelle to talk about the market.
Thank you, Julian. Based on the very encouraging clinical data that Julian has just shared with us, we have had extensive discussions with transplanters in the United States, which would be our first potential regulatory approval. And what we have learned through both live discussions and blinded market research is a very good understanding of the overall market for allogeneic stem cell transplants, a very good understanding of the Meduvacell potential, and then I will also walk you through our plans to achieve the goals that I will discuss upon FDA approval and launch. So when we look at the market opportunity in the U.S., there are over 40,000 patients each year with hematologic malignancies who are considering transplant. The opportunities for Meduvacell fall into 2 key categories. One is expanding access, and the other is improving outcomes. Let me walk you through each one. So by expanding access, I want to focus on those patients that are deemed eligible but do not go to transplant. That patient population is estimated to be about 9,000 patients each year. One of the key reasons that a patient could be deemed eligible but not go to transplant is unfortunately they are not able to find a donor, similar to Stacy, who Julian had introduced you to prior. This is devastating for the patient, for their loved ones, and for the transplant center. Once a patient is diagnosed with an acute leukemia or an aggressive lymphoma, generally their only path to a cure is transplant. So in this case, when the patient is not able to find a donor, it is very challenging. Meduvacell, because we start with cord blood at the beginning of our manufacturing process, we have a less stringent matching criteria. So hence we have the ability to match those patients who may not have been able to potentially find a donor. So that's the one opportunity in terms of expanding access. The other key opportunity is improving outcomes. So there's approximately 10,000 patients in the United States each year who are transplanted. And when physicians look at the clinical profile of Meduvacell, what they reference in terms of our ability to improve outcomes are both the efficacy data, specifically the primary endpoint of time to neutrophil engraftment, but also some of the secondary endpoints, reduction in infections, reduction in time in the hospital. The other key factor that physicians bring up quite a bit is the age of the donor. Age of the donor has direct impact on patient outcome. The higher the donor age, the worse the patient's outcome is. And in the case of Meduvacell, because our starting material is cord blood, and because the cord blood is derived from an infant, the age of the donor is not a concern when a patient could potentially be dosed with Meduvacell. Let me now explain in terms of patient numbers what those two opportunities would equate to. Upon achieving peak market share in the US, which we anticipated would be approximately 3 years from launch, we see the opportunity to potentially treat approximately 2,400 patients each year with Meduvacell. Those patients would be roughly divided evenly between the opportunity to expand access and the opportunity to improve outcomes. We do anticipate that number of patients treated each year could potentially grow. It could potentially grow as just the size of the US population grows, but also as advances move forward for the treatment of acute leukemias or aggressive lymphomas, more patients could potentially become eligible for transplant. We are very excited about this opportunity. So now our important step forward is how do we assure launch readiness upon potential FDA approval? First and foremost, our key focus for the launch will be assuring a positive patient experience. Patients who would potentially be candidates for Meduvacell have been through so much already. The devastating diagnosis of an acute leukemia or an aggressive lymphoma, the journey to go to transplant. We want to make sure we are developing our launch approach with keeping the patient at the forefront, at the center of everything we are focused on. Our launch tactics and strategy will focus on 4 key areas: educating the transplant centers in the United States, educating the payers or the insurance companies, having that great support group, which we will refer to as Gimida-Cell Assist, to provide support for patients in hospitals, and then focusing on our commercial manufacturing readiness. So let me touch on the transplant centers and manufacturing in a bit more detail. In the United States, there are 70 transplant centers that make up about 80% of the transplants. The exciting opportunity for Gimida-Cell is roughly a third or 20 of those centers already have had clinical trial experience with Meduvacell. We know those centers well. We also recognize that we could very efficiently launch Meduvacell in the United States. We anticipate a field force of approximately 25 commercial account managers and probably less than 10 medical science liaisons. So we feel not only we know the path forward to educate the transplant centers, but we could do so very efficiently. And then in conclusion, I would like to wrap up with our progress on manufacturing readiness. And let's start right here in Israel. We have an outstanding facility that was built in Kiryat. Construction was completed middle of 2020. Our initial operator team was hired, they were qualified, and now we are underway to fulfill the requirements for our BLA as discussed with the FDA. And we're not only focused on the BLA requirements, we're focused on getting ready for launch. We need to make sure upon FDA approval that we are ready for launch. So the manufacturing team in Kiryat has done an outstanding job not only focusing on BLA readiness, but preparing for launch. We also have a secondary manufacturing site that we will be submitting for commercial manufacturing, and that's a partnership with Lonza, a very well-recognized global company focused in a number of areas, including cell and gene therapy, so we are partnered with Lonza as a secondary source. So in conclusion, for Meduvacell, we are very encouraged by the clinical data. We have received very good feedback from transplant centers, from payers, about that clinical data and the impact it could potentially have for patients and both expanding access for patients to get to transplant, but also the potential for Meduvacell to improve outcomes. We not only have a clear understanding of the market, a clear understanding of the Meduvacell opportunity, we are now well underway with our launch readiness plans. So let me turn the discussion back to Julian to now introduce and walk us through our natural killer cell platform.
Thank you, Michelle. GDA201 represents an expanded natural killer cell source to treat cancer. Natural killer cells is the first line of defense of our immune system, and every day natural killer cells protect us from infections. However, in cancer patients, the natural killer cells unfortunately are not a sufficient robust number of cells and become exhausted, become ineffective for cancer patients. The benefit of natural killer cells is that we can get those cells from young donors, young healthy donors. There's no genetic matching requirement. These are innate immune cells, so no HLA requirement at all. And as you can see from the cartoon, there's synergy with antibodies. Based on the expression of CD16 on the natural killer cells, they bind the tail or the Fc portion of antibodies to enhance the killing effect. And the antibodies target the tumor antigen. So in the case of lymphoma, it's anti-CD20, and there's a very well-known antibody called rituximab or Rituxan, which can target the tumor cell, and as you can see from the cartoon, the NK cells come in on the back end and enhance killing by a process called ADCC, or antibody-dependent cellular cytotoxicity. And our expansion is able to take from a single donor and make billions of cells, many billions of cells, and many doses from a single donor. Our first in-human study was done at the University of Minnesota under the direction of Jeff Miller, and Dr. Veronica Bakanova was our principal investigator. This was an investigator-sponsored study where our NK cells, we transferred the protocol to manufacture them to Minnesota, and they used fresh cells at the University of Minnesota to treat patients either with non-Hodgkin lymphoma, NHL, or multiple myeloma. And we paired the antibody depending on the disease. So as I mentioned, Rituxan for non-Hodgkin lymphoma, as we saw in the previous cartoon, or for multiple myeloma, elotuzumab or Implicity to treat myeloma. All patients got a loading dose of the antibody, then a light dose of lymphodepleting chemotherapy to make room for the NK cells. Then they got GDA201 by a single infusion, and a subsequent single dose of antibody. I'm going to focus our discussion today on non-Hodgkin lymphoma because of the nature of the antibody in myeloma, the results were not as encouraging. We evaluated 3 doses in an escalating dose manner, and I'll show you those doses in a moment, to evaluate both safety and overall response rate. In the lymphoma cohort, we treated 19 patients, and we were absolutely amazed to see 14 of 19, or 74% overall response rate. And amazingly, 13 of the 14 patients had a complete remission, complete response, no evidence of disease. And they were split by 2 lymphoma histologies, follicular lymphoma, which is a more indolent, slow-growing lymphoma, or the very aggressive lymphomas, diffuse large B-cell lymphoma, DLBCL. If we look at overall patient safety, we included the myeloma patients as well, and we have 35 patients evaluated. We had no dose-limiting toxicities. We had 1 patient in the myeloma cohort who came in with an infection which was not diagnosed and unfortunately developed sepsis and died. But the most common adverse events of high grade were related to the lymphodepleting chemotherapy, and they were transient and mostly asymptomatic. Importantly, unlike in the CAR-T field, we saw no neurotoxic events, no graft versus host disease, or no confirmed cytokine release syndrome or CRS. So very, very well tolerated, and potentially an outpatient treatment. Shown here is a swimmer's plot where every line represents a patient. So all 19 patients are indicated here. And we examined 3 doses. The lowest dose was 20 million cells per kilo, the mid dose was 100 million cells per kilo, and the high dose was 200 million cells per kilo. The majority of these patients were in 3rd and 4th line, heavily pretreated, and had failed multiple Rituxan-containing regimens. The majority of these patients also had a very poor performance status, a Karnofsky score of less than 80, and they were not eligible for CAR T treatment based on their health status. We saw responses at all doses with follicular lymphoma. Almost all patients responded. But for DLBCL, the more aggressive form of lymphoma, we saw only responses at the mid dose and the high dose. And we settled that the high dose would be our recommended phase 2 dose. Now, what's very interesting is the NK cells only persist for about 7 to 10 days. So they're gone from the body with less than 2 weeks. And if we look, for example, at patient 009, the third bar from the top, initially this patient had diffuse large B-cell lymphoma with a partial response, shown in yellow. But that response improved over time, long after the NK cells had departed the body. And this continued improvement turned into a complete response almost a year after treatment. So what this means clinically is that the initial innate immune response and the ADCC provided by the NK cells combined with rituximab triggered the adaptive immune response, which recruited CD8 and CD4 cells and created what is potentially permanent immunity to the tumor. And you can see from the time of treatment, this patient is over 2.5 years from their treatment and is disease-free. You can see there's a lot of green on the slide, and our working hypothesis is being proved now with biopsies to show that in the body we are actually triggering the adaptive immune response. So let me show you a picture, because this says the whole thing in one simple picture. This is that patient 009, a gentleman with 57-year-old initial treatment of chronic lymphocytic leukemia, which transformed into Richter's transformation in a diffuse large B-cell lymphoma phenotype. You can see the prior number of therapies, many lines of therapy, and progressed on all these lines of therapy. This gentleman was fortunate to have a sibling match and did get a hematopoietic stem cell transplant, a bone marrow transplant, but unfortunately relapsed after 6 months. So this patient had no treatment options and was not a candidate for CAR T treatment. So came onto our trial as the last resort, and as you can see from the images, particularly looking at the baseline images at the top, I direct you to the heavy burden of disease in the abdomen, and after 6 months, almost all the disease was eliminated. There's a little bit of the tracer in the heart and in the bladder. That's very common because the tracer accumulates in the heart and is eliminated in the bladder. But there's really no evidence of disease. So to conclude, we believe that our key accomplishments is we have a very clear and dramatic proof of concept. And this has given us confidence to develop this cryopreserved formulation, which we have successfully done in our laboratories in Israel. We are now in a GMP facility. We have completed the engineering runs and the GMP runs to put product on stability and create inventory to support what will be a phase 1/2 multi-centered study, which could support, if positive, an accelerated approval approach and registration with the FDA. Future directions, as I've discussed in my opening remarks, is that we've been able now to learn how to genetically modify these cells using both CRISPR technology, gene editing technology, as well as chimeric antigen receptors, CARring these cells to address solid tumor cancers, which are much more difficult to treat because of the immunosuppressive microenvironment of these cancers. So in conclusion, I end where I began. We have 2 clinically very promising candidates and are advancing those candidates with Meduvacell, poised to be filing the BLA by the end of the year and poised to be reviewed by the FDA. And with GDA201, with the cryopreserved program, we are on track to submit the IND this quarter and to initiate clinical trials by the end of the year and to begin treating patients with non-Hodgkin lymphoma. And again, I reiterate our strong financial position, and I thank you all for joining us on this webinar. But most importantly, I would like to thank the patients and their families who participated in our clinical trials, as well as the caregivers who treated them. And finally, and not least, is I would love to thank the employees of Gamida-Sel who worked through this COVID period, who worked tirelessly to try to advance our programs because we are focused on trying to cure patients with these devastating diseases.
Julia and Michelle, thank you very much for a great presentation. Shai, thank you so much for joining us. From now, we're going to take it to the questions, and we'll start with you, Michelle. We're talking that we expect a certain pace of penetration with the OMI product. Would you please try to define us how will it work? What is the concept right now on the treatment with this relevant problem?
For Meduvacell in the United States alone, there's really two key opportunities for penetration and for patient opportunities. The first is to expand access for those patients, especially those that are deemed eligible but do not go to transplant. In the United States, there's approximately 9,000 patients each year that are deemed eligible and do not go to transplant. Because Meduvacell has a less stringent matching criteria, we have that ability to potentially help those patients. The second key opportunity is to improve outcomes. In the United States, there's approximately 10,000 patients transplanted each year, and physicians have shared with us when they look at the Meduvacell clinical data, the efficacy data, specifically time to neutrophil engraftment, and then the secondary endpoints and the safety data, that Meduvacell has the potential to improve outcomes for patients who are currently getting transplanted by other modalities. So those two opportunities, expanding access and improving outcomes, what that equates to in terms of patient numbers is by the time of peak market share, which we anticipate to be about 3 years from launch, we would expect approximately 2,400 patients treated each year with Meduvacell. That number has the potential to grow because the U.S. population will grow, but also we will see an increase in number of patients who are eligible for transplant as more advances are made to get patients into remission. This is a very encouraging opportunity for Meduvacell, and we are excited and ready for our launch readiness plans.
Thanks, Michelle. This one maybe to you, Julian. What do you see are the risks for your product and for your model, and how are you planning to address them?
Sure. Well, for Meduvacell, the major risk, the clinical risk, has been resolved because we have positive phase 3 data. We met our primary and our secondary endpoints. We've also shared those data with the FDA, and they've agreed and reviewed the top-line data. And by the way, we just published in Blood yesterday the full manuscript of the phase 3 program. So the risk is really now all technical. It's the preparation of all of the documents needed for a BLA and all of the activities required to manufacturing. For cell therapies, the manufacturing site has to be inspected by the FDA. And our new site in Kiryat Gat, just south of Tel Aviv, will be physically inspected by the FDA. So we're getting ready for the Israeli Ministry of Health audit. And then we're going to be also getting ready to treat patients from that site to file to the BLA to show clinical comparability.
Thanks, Julian. We see a lot of PDUFAs and approvals delays over the past few months. Are you scared this might happen to OMI's approval?
Very unlikely. I will remind you again that we have FDA breakthrough therapy designation as well as orphan drug status. And as a result, we are at the front of the FDA list to be reviewed. And based on that, we will ask for priority review, which means a PDUFA date of 6 months, up to 6 months. And so we will get a very rapid and high attention from the FDA.
Which is different from the PDUFAs given to other companies? Okay.
Standard review is about 10 months.
Okay, thank you. How would you take the strategy of the pricing of the Meduvacell product? How do you do it? How do you usually look at it? Maybe as much as you can elaborate on this topic, I think we'll appreciate. Michelle, maybe?
So we will not guide or announce price until we have FDA approval. What I can share at this point in time is some very important feedback we've received from the United States payers or insurance companies. The payers are very encouraged by the clinical profile of Meduvacell, but they're also encouraged by the reduction in healthcare resource utilization. For example, as Julian has presented and as we are published in the recent journal article in Blood, we have a statistically reduced time in hospitals for patients. That then translates into a reduction in healthcare resource utilization. So in addition to our clinical data, we are also preparing an extensive health economic analysis that will be shared with United States payers or insurance companies. You know, we do sometimes have discussions with payers, and when they bring up potential pricing analogs, and there's no perfect analog for Meduvacell, it's a very innovative therapy, a unique therapy. Sometimes payers do bring up other cell therapies, though, and the most recent cell therapies to not only receive FDA approval in the United States, but also to launch and get reimbursed are the CAR T therapies. And we know the CAR T therapies in the United States were priced ranging from approximately $370,000 up to approximately $470,000. We look forward to continuing to partner with the United States payers for not only the clinical discussions, but also the health economic analyses.
Thanks, Michelle. Maybe I'll ask you again, Julian. Why not go into a partnership deal with a bigger firm? It's a marketing issue. I'm not sure that Gamida-Cell, which was always an R&D company, should focus on the marketing. Maybe you can explain why you decided to go that way.
Yeah, thank you for your question. Obviously, that's kind of the standard for a biotech company. However, since you saw that this is such an orphan indication, where the very concentrated footprint, Michelle pointed out that the top 70 transplant centers account for 80% of the procedures. We believe that our commercial team can be 30 to 40 people, and in a very concentrated accounts, that's something that's very manageable for a company our size. So that's in the US. But looking at other geographies like Europe or Japan, we are considering licensing to regional companies to cover the global market.
Thanks. Do you see any further competition to your segment in the future? Maybe not in the close one. Other clinical trials that you see going on that might risk your novelty in the market?
So we are the most advanced. We're the first company to ever run a randomized phase 3 study in bone marrow transplantation. There were several other competitors. Several have abandoned their programs once they saw our phase 3 data.
Can you give us names, maybe?
Well, Magenta was, which is another Boston-based company, had a different expansion technology. Was not as robust as ours. The time to engraftment was also not as robust as ours. When they saw our 10-day time to engraftment, they subsequently abandoned their program. And so I think we don't really have any competition right now. And based on our profile, I think we have, we are the market makers.
Michelle, how will you approach the hiring of your commercial team for Meduvacell?
We are hiring very experienced individuals in hematology, transplant, and cell therapy. I had the very exciting opportunity to launch a cell therapy in the United States when I was part of Kite Pharma, and we launched our CAR-T in the US market. So I have a very good understanding of the importance of hiring experienced people in order to appropriately launch cell therapies. So in the case of Meduvacell, that is the profile we will follow also. So we have our leadership team in place already on the commercial side. And in terms of the field presence, it will be a very efficient launch. We will have approximately 25 commercial account managers partnered with approximately 10 medical liaisons or medical personnel in the field. So we are confident that we're not only hiring the right people, but we could do so very efficiently in terms of a very efficient size of our field team.
Another shorter answer to that question is Michelle's our secret weapon.
Maybe later we will elaborate more about Michelle's prior achievements. I want to move to your NK product for the moment. So maybe you, Julian, please, what is the new hype around NK cells? What's so exciting about this?
In the last 5 years, CAR-Ts have been around. The whole field of cell therapy has kind of shifted to understanding NK biology, natural killer cell biology. It's very early days. No one's beyond phase 1, but it's the hottest field in cell therapy for cancer today. And there are many, many companies actually pursuing different kinds of technologies. What differentiates us is we have our cell expansion technology, is that we can get those cells from normal volunteers and just expand and make 50 to 100 billion cells in 2 weeks. So that's our contribution, and the nature of our expansion gives us the phenotype of those NK cells so that they have a memory and the expression of CD16 and the homing capacity to go to the right tissues where the tumor is.
Okay, thank you. Maybe a timeline or a horizon that we can see for this product to become a product that is being used on and approved by the FDA?
So first is manufacturing. And we've successfully been able to manufacture these cells in a GMP facility. But equally importantly, and NK cells are very challenging to cryopreserve, to freeze the cells, because when you thaw them, they lose their activity very, very fast. They're very delicate cells. But our scientists at Gamida-Cell have solved all these problems. We can recover almost 90% of these cells after thawing. So having this cryopreserved formulation, which will be off the shelf, enables us now to build an inventory of many bags of cells so we can support a multi-centered study.
If you say that it was 90% that you were able to preserve, what is the normal number that?
When we began, it was about 50% recovery, but now I should have said over 90%. I said almost 90%, but it's really very, very high potency. And we've tested the fresh versus the cryopreserved, and we've maintained the potency. And we have lots of tests in the lab to observe that potency.
So for this product, when do you think you can show any maybe relevant deals with partners? What are the type of partners that we're looking for right now?
It's an interesting business question, because there are two types of partnerships that we can consider. As I also told you, we're also engineering these cells, and we have the CRISPR-Cas9. We have great capabilities to be able to engineer these cells for specific properties. So one can envision a big partnership, a comprehensive partnership with a big pharma, and we've seen deals like that with, for example, Fate Therapeutics and J&J, or Sanofi that acquired the Kiadis technologies. So we are maybe next in line to consider such a deal. Alternatively, we could decide to keep GDA201 for lymphoma and then partner with individual companies for individual targets that fits in their pipeline portfolio of indications. So we're exploring both of those actively, and we have a lot of inbound requests as we've presented our data publicly, what you've seen today, and so the phone rings and they call us as well.
I think we're going to ask some general questions, maybe more about the finance, so we're going to go to Shai with your, if it's okay with you. So we're seeing a burn rate of almost $120 million per year. Should we expect the same pace?
First of all, hi everyone. Thank you I'm very happy to be here. As for your question, I can say that nothing significant changed versus our public guidance we made earlier this year. We are going to burn from operating activities around $110 to $120 million this year. As for 2022, of course, we are not in the position to provide any future guidance these days. Having said so, I think it's reasonable to assume that as we get closer to launch, the burn rate will be higher. However, just to remind everyone that potentially a year from now, we expect to see some initial revenues from Meduvacell that will offset this burn rate.
So, assuming you're getting closer to the launch, are we expecting higher costs? Where are we supposed to have the money from? You're going to raise?
So thank you for the question. These are very valid questions. As you all know, there are multiple ways to finance the company, including, as Julian just mentioned, strategic deals and ex-US partnerships and some others, strategic transactions. Of course, we do not comment on any specific activities that we are doing these days. Having said so, I can assure you that we in Gmida are always evaluating all options to bring value to the company and drive our program forwards. Just to remind everyone that based on our recent financing we did in December last year and the iBridge deal that we did in Q1 this year, we have sufficient cash through a very, very significant milestones, including the BLA submission for Meduvacell and potential approval in less than a year, and submitting our IND for cryopreserved GDA201 program.
So we're going to see a raise on the burn rate for sure. We're going to see also some revenue starting moving on. So can you please maybe close us on the IP that covers the technology, the NAM technology, and your product so far?
Our IP has 3 pillars. First of all, we have orphan status. So we have 12 years exclusivity in the US for a BLA, or a biologic product. We have 10 years exclusivity in Europe at the time of approval. Secondly, we don't have inventory with Meduvacell. Each patient that needs Meduvacell, we manufacture for that patient, and within 4 weeks, they consume the product. So there's no inventory, which means you can't make a biosimilar. Lastly, we also have trade secrets. And those we keep private. So it's impossible for someone to reverse engineer what we've done without doing a full clinical program, which took many, many years and many millions of dollars. And we do have IP, though. I don't want to neglect that. We have methods patents, methods of manufacturing, methods of treatment, and our youngest patent takes us to 2038. So we think we have a very strong, comprehensive exclusivity period.
Thank you. What do you see as the key milestone for the next 12 months or so? I think I'll ask that. Okay. Julian.
The key catalyst will be filing the IND for GDA201.
Expected
in the coming months, because we've already successfully manufactured, put product on stability, and we're building inventory. Every day we're building more product to be able to distribute to patients. So we file the IND. There's usually a 30-day FDA review, and then we can open up sites. We've hired a global CRO to support multi-centered studies. And we've already had advisory boards with key clinicians, world experts in lymphoma, and are selecting our sites and our investigators, and we're in constant discussion with them. So the hope is that we will treat our first patient before the end of the year and into next year. And the trial will begin with requalifying phase 1, the dose that we got in Minnesota, 200 million cells per kilo. We will requalify that dose for safety and then move into the phase 2 portion where we will treat follicular lymphoma patients and diffuse large B-cell lymphoma patients. The other major milestone is the pre-BLA meeting with the FDA to set up the BLA filing by the end of the year, and that will put us on under review. So very busy next 6 months of 2021, but it's all execution. It's all execution, and our employees are so dedicated, so focused on meeting these milestones.
It seems like you're in the last mile for OMI and for many other products here. I want to thank you all.